and welcome back from lunch. It's my pleasure to introduce the second session of the day focused on the impact of childhood adversity. I've been asked to keep things pretty quick and move us along at a good pace so that everyone can get to the talks on time this afternoon. So this will be another nicely interdisciplinary section. We'll draw on faculty from psychology, biobehavioral health, and human development and family studies. And so specifically, our first speaker for this afternoon session is Dr. Martha Wadsworth, who's been in the psych department here since 2012 when she came from Denver. Is an associate professor there and is the director of the Coping and Regulation of Environmental Stress Lab. And she'll talk to us today about translating a commitment to social justice into biologically informed prevention. Thank you.
Thanks Jenny and Emma for inviting us to do this. It's been really fun listening to all the talks so far. And so I'm going to tell you about my own adventures in translational science. And going back to the beginning. What? Oh! She did tell me to turn it on and I forgot already. Okay. I'm just going to tell you about my research program, which has really gone back and forth between basic research and applied intervention work, and then back to basic research, and then back to intervention, and back to basic again, as I use the results of intervention research to then learn more basic science questions, and then of course use those to inform interventions. But there's always a central kind of core theme that I got from my undergraduate advisor, believe it or not, who was George Albee, and he was a radical community psychologist, and his ideas, he was considered by many the father of primary prevention. He was a clinical psychologist, but he was very, very adamant about the fact that treatments are never going to reduce incidence rates of mental or physical disorders. That really you have to prevent them from occurring in the first place. And the radical piece was that you really have to attack the causes of the causes, not just the mechanisms, but that we have to really address issues of power inequality in our society. Grand vision. But he also said, and use your best science to do that. So my research program has really been trying to stay true to the social justice roots of George, but do good prevention science as well. There's strange things up here. Okay, so the thing I am most passionate about is kids in poverty and the negative effects that poverty has on their development, both physical health and mental health, in particular, health problems. And how do we get from kids exposed to economic injustice in our society to healthy kids? The pathway looks probably something like that, not terribly straight. And as it turns out, that's kind of been my trajectory too, sort of the basic to the intervention, back to basics. So when I got started in the field, one of the prominent models of how exposure to poverty affects kids' development was a stress-based model, the family stress model put out by Rand Conger and Glenn Elder. And they really, what I liked about the model was that these things that we like to call deficits in our field, parenting problems, oh God, I can't see, parental psychopathology, interparental conflict and violence in the home, et cetera, they situated those as a result of the stress stemming from things like poverty. So it wasn't inherent to the people. They were not bad parents. They were not sick people, but that their problems were stemming from these structural problems with our society. So I really liked that. It fit with my philosophical bent. But the kids are like nowhere in that model. They're only at the end. So my question was, what about the kiddos? Do they experience economic strain, or where does stress fit into that on the kids' side? So that's sort of been what I've been pursuing ever since. And I was also really interested in where the heck are the protective factors. This was a total deficit model, and I'm not a big fan of deficit models. I want to write a chapter on dump the deficit model, but we'll see. Nobody will publish that. Okay, so I started, with a bunch of burning questions. The first being, do kids experience economic strain? That's at the beginning of the model, and we know it affects parents, but does it affect kids directly? I don't know. People said, nah, they don't. I'm like, well, as someone who grew up in poverty, my 6 siblings and I certainly felt the stress of it, but that's only an N of 7. So I went out to ask the question of a few more kids. And so for my dissertation project, I got a sample of 400 kiddos and their parents, and got really detailed information about what kinds of things happen in their lives that cause them stress, that make them feel bad. And then I've been examining, how that is related to various types of outcomes, physical health, mental health outcomes. And one of the sort of first things we discovered was it's not just financial stress, not just not having enough money, but it was all these other things that go along with it. Exposure to violence and conflict in the home and out in the neighborhood, and maltreatment and all kinds of things, right? All those things that were on Denny's first slide and are co-occurring on other slides. And so I came up with the term poverty-related stress to really capture all the different types of negative things that the kids are having to cope with when they are living in poverty. And then started reading about, oh gosh, there's a biology of stress too. So I started really attending to some of these newer models of toxic stress and allostatic load, and that became part of my understanding, and that's where biology started coming into the way we were thinking about things. So, okay, not a fan of deficit models. So, okay, got it, check. Economic strain, kids experience it, and the more they have, the worse off their functioning is. What about some protective factors? Given that stress is so critical to these models, might their coping make a difference? Again, empirical question. So we designed some studies to really look at that. I kept finding this intriguing and confusing finding that it came up in my dissertation sample, and then it came up in my next longitudinal sample, where kids exposed to higher levels of poverty-related stress had the most maladaptive coping profiles. And so rather than using a lot of active problem solving, et cetera, they were using a ton of avoidance and aggressive coping, and they were hypervigilant, and we're like, what's going on here? And coping, positive coping, problem-solving, active coping, still predicted better outcomes. So it was this weird mediation where poverty was predicting worse coping, but good coping when they used it was related to good outcomes. So we're like, huh. And we started thinking about, wait, what's going on here? I think that's about the time I started reading about adaptive calibration models and started thinking about, maybe these are not just deficits. Why do kids develop hypervigilant, avoidant, aggressive coping styles if they grow up in poverty, exposed to a lot of poverty-related stress? Maybe it's adaptive. Maybe it keeps them safe in dangerous environments, to be vigilant to everything, to punch first and ask questions later. I know it doesn't work well in the classroom, but it might work well in the neighborhood if you're being hassled by a drug dealer, et cetera. And the kids that, I've worked with increasingly high-risk kids, and these are the situations they're in all the time. So those we've come to understand it as functional adaptation. These functionally adapted coping repertoires keep them safe. So when we think about intervention, do we really want to get rid of those abilities unless we're going to magically transform them out of those neighborhoods. So this is all percolating in my mind as I'm thinking about, okay, I am a clinical psychologist. I really want to do something to help, right? So how do I use this information to design powerful interventions? So what if we teach active coping to these kiddos, not unteach the other stuff, but what if we add skills on top of what they have? And so we developed a pretty brief coping intervention for kids exposed to poverty-related stress. The Families Coping with Economic Strain program, and it showed a short-term effects on preventing the development of internalizing problems. Whoops, did I just do that? Never mind, okay. And so it was good for coping with certain types of stress. But then it was not so helpful because we were teaching problem solving, cognitive restructuring, relaxation, et cetera. We didn't have a lot of tools to give to the kids when they were talking about their parents beating each other up at home or being hassled by a drug dealer on the street. And we're like, oh, yeah, take some deep breaths. That seemed really insufficient. So we had promising effects, but really suspected that was not going to be the end-all, be-all, or have an effect at the physiologic level. So we went back to the Conger model and said, what about the parents? Maybe we'll get more bang for the buck if we actually work with the parents, right? Yay, parents are great. So we recruited a huge sample for this kind of research. 300 low-income mothers and fathers and a child. And we developed an intervention specifically for them based on the family stress model. We targeted the 3 pillars, the 3 things in that first model. Stress, so we taught them stress management and coping skills. Interparental conflict, we taught them communication, conflict resolution, effective sort of family parenting. Oh, and then the third is parenting. We worked on co-parenting as well as regular effective parenting skills. It was great. And so the idea was that they would have, by strengthening the family unit, there would be these positive downstream effects on kids. Turns out, 50 to 60% of the parents in our study were not really able to uptake these kinds of skills. They were coming in asking for money for diapers and for food to feed their families. We had recruited, an amazing sample of very poor families, and they were just not in a place to be able to learn the skills. They needed a different kind of intervention. They needed a systems level intervention, right? Okay, back to the drawing board again. So what about all this new research involving the damaged stress response systems? And maybe we should learn more about what that's doing, how that is associated with poverty-related stress, and what we might learn from that to design our most potent intervention that we possibly can. So, and what will it take to affect this physiologic change? And then also, remember, I cut my teeth with a radical community psychologist. I can't let go of the fact that structural inequality, marginalization, discrimination is at the core of what's happening in these kids' lives. So I went back and did a study funded by NICHD looking at the physiologic signatures of different types of coping. So we had kids come into the lab and we put them through stress induction and randomly assigned them to cope in different ways. And then we looked at their ability to downregulate physiologically after the stress in the different coping conditions. And so we identified some better and worse ways of coping at the physiologic level. So we're like, ooh, okay, this has promise for intervention. And we found out some more stuff too, but I'm probably getting close to being out of time. But so we found, yes, physiology seems to change with coping. And this was an hour and a half this wasn't even like, 16 hours of intervention. This was just one little segment of time, and we showed movement. So that was very cool and very encouraging. So as we're thinking about adding onto that original FACES intervention, which we, had good effects. They learned fundamental, regular old coping skills, but now we want to add pieces that will potentially have potency at the biological level. So what are some resources that kids in poverty and minority youth and working class youth might have that we can really build on to scaffold a different type of coping? Because coping with what we call societal stress, racism, discrimination, 5 minutes, okay, requires something different. You can't do it alone. And in fact, we tell that, you can't do it alone. You can't go out and solve the dangerous elements in the neighborhood by yourself. So what do? Do you just turtle and stay calm and cope with a crappy situation and move on? No. Hell no. Social justice, no way. So we go back to the empowerment literature, the social justice literature, and they suggest that we need to give these kids a voice. Their voices need to be heard, and give them a way to be transformative, give them kind of transformative experiences so that they can take action to deal with these things, not just stay calm in the face of adversity. So we got into social identity complexity. So the ethnic identity and racial identity literatures show real strength in those, that kids who have a strong sense of who they are and have strong connections with their racial and ethnic backgrounds seem to be better able to handle stress, and it buffers them against some of these stressors. So as a sociologist at heart, I think, I started looking into working class identities and the positive aspects of working class and working poor identities, which we tend to, in our culture, denigrate, right? Poor people are stupid and lazy and bad. But think about the working dude. They're hardworking, they are persistent, they're loyal. And if you dig into it, there's a ton of strengths there. We just don't value them in our everyday culture. But there's a ton of strength to be found there if we give kids a place and a way to validate those and draw strength from it. So the intervention we're currently delivering and evaluating now incorporates all of the pieces that we've been talking about. So we still teach them fundamental coping skills because you need them to do anything else. And then we have them really try to understand who they are, who they're connected to. We do this really cool activity of a tree, and we give them a big poster of a tree, and we have them attend to and discover who their roots are, the people and the practices and the rituals and the families and the community events that nourish them. And then they are the branches, and we have them explore who they are and who they got those characteristics from. Focusing on things like, I'm a hard worker, I'm kind, I'm loyal, and really, because again, kids in poverty in our culture get a message that they're deficient, they have deficits, they are stupid, bad, lazy, etc. And what we're doing is really helping them connect with their strengths, their indigenous strengths, if you will. And so with fundamental coping blocks and a strong kind of sense of who they are and how they're strong, we then teach them about social action as a way of coping with the bigger societal ills. So while you may not be able to do it alone, you can join with other like-minded individuals and do things together to make things better. So the final portion of our intervention is a community project where the kiddos actually take ownership of it and design and implement a project of some sort to make their little corner of the world better in some way, prettier, safer, whatever. We've had a variety of things from, one group adopted a vacant lot in Harrisburg for $1 a year. We get to rent it, and they cleaned it up and turned it into a park. They painted benches and put birdhouses in, et cetera, as a way to create a little more green space in their community and a safe place for kids to play. Our current group is so cool. I love what they're doing. One of the modules in the middle of the intervention, they create coping toolboxes. So they make stress balls, and they make little cards to themselves about what are the different steps of problem solving. And so they're able to take their little box. So the current group of kids that we're running right now, their community action project is to create coping toolboxes for all the kids in their class at their school. We're at Steelton Elementary School. And so they created 150 coping toolboxes, just the 9 kids in the group. And they're spreading better coping and better self-regulation, hopefully, throughout their community, which I thought was a super creative and kind of neat community project that I couldn't have come up with. That required the kiddos and what they bring. Okay, the other piece. So we're trying to affect the HPA and the SNS. So we're trying to affect the stress physiology. We're trying to kind of repair the stress physiology. And so in our pilot work, we have found movement on that at post. A lot of our kids come in with very blunted levels of cortisol, showing really high risk physiologically. And at post, they actually have a more typical response to stress in the lab physiologically. So there's promising results. We're doing an RCT now to hopefully show it on a larger scale. So we think we have some ideas for how to grow healthy kids. We always want to grow more healthy kids, more, bigger, better. And that's my next project I don't have time to tell you about, but we're gonna hopefully learn more about how to protect a wider variety of things other than just the physiologic stress response system. And so that's the basic to intervention to basic to intervention and back again trajectory that I've been on. And so just, I think, similar to some of the messages in Denny's talk and even in some of the discussion we just had, that really to meet the needs of kids in poverty is going to take a variety of approaches, including, structural, dealing with structural inequality. Because in the end, if these kids were not poor, we wouldn't be having this conversation. In addition, I think for a lot of parents that we want to work with, they're not ready for the kinds of interventions that we have that could be very helpful. But if they're still struggling to put food on the table, it's irrelevant, right? So we really need to do a better job, I think, of pairing up more systems level interventions with the kinds of things we're coming up with. And in the end, we're trying to empower kids with skills to transform the world and keep their brains healthy. Small thing. Yeah. And just we're working in a variety of ways with school districts and other agencies to really try to help those agencies and school systems to do a better job of selecting evidence-based programs to implement in their schools. You'd be amazed at some of the garbage that they pay a lot of money for, and you're like, there's no hope in hell this is going to do anything. So really helping them evaluate the evidence and implement more evidence-based practices. I think that's good. Thanks. Thanks for putting up with my too long.
I think we're just starting the meeting, but I think we'll be up and running again in a second. So in the meantime, let me introduce our second speaker. So Lisa Gatsky-Kopp is an associate professor in human development and family studies here at Penn State. And she's been here since 2008, I believe, and leads the Child Brain Development Lab. And we'll be talking about adaptations, compromise, which you can't see, but we'll hear everything about in the next 20 minutes or so.
Indeed. So I will take this moment to say, as a full caveat here, that I was not originally on the schedule to speak and was asked to step in at a late notice when somebody canceled at the last minute. So please take that into account when you judge me. 4, yes. So this is just trying to, do a good thing here, and it may not be the most detailed scientific review of my research. And actually, I kind of looked at Denny's instructions about what they were going for and thinking about translational implications and policy implications. And so I took that kind of notion with the talk of what would I say are the things that we should be investing in as a community, as a society, based on the science that we have right now. And a lot of this is at a much broader level about the kind of theoretical models and theoretical understandings of development that guide my work, more than the specific findings from my work. I'll just sort of point out, like, I do some of that, but that's not going to really review my findings so much as basically reach the conclusion that if I had all the money that Congress had, I wouldn't spend it on my research. I'd spend it on a few other things that are probably more important. But I think that my talk will fit in nicely with the kind of other talks that people have been giving today, and in particular, this idea of whether or not developmental changes that are happening in the context of adversity are really adaptations, and what does that mean for our understanding of development. So I am going to, I guess, get started, and here's this thing. Okay, yeah. So you got my kind of overview. This is just a general model that I like to use a lot in when I'm teaching and talking. This is a basic Gottlieb's model of different ways to think about different levels of analysis when we're talking about development. So there's this idea that we have, we can study the genetics, that the genetics are influencing neural activity, which has an effect on behavior. Behavior goes out into the environment, and that all of these things are kind of cascading back across time to shape each other and have influences on how individuals. Okay, so
Tell them to mute.
hello, we can hear you.
11:37, it said something like,
David, can you mute?
what?
Mute.
Oh, sorry.
That's okay, you give the talk. You give the talk, it's cool.
I had to test before I Sorry about that. Thank you, sorry.
No problem. So what I think is interesting about this is sort of, it gives you an idea of where might we want to intervene and see different effects, but it also, it kind of gives this fits with the basic notion we have in development about this idea of that there are temperamental qualities, right? This genetic and neural underpinning of behavior that maybe is happening at one level of analysis. And when we do most of our interventions, we're at that behavioral and environment kind of level. And that can lead us to think about things like person-environment match, or personalized medicine, tailored interventions, and those kinds of things. But back in 2004, I actually was at the Society for Neuroscience convention, and I wandered into a talk by Michael Meany, which was a little bit life-changing for me. And it really upends this model for me in a very simple but profound way, and that is I'm looking at a wrong screen. In thinking about moving that environment down to the bottom, right? That these things that we think of as immutable characteristics, genes, neurobiology, those aren't just traits that arise and we have to deal with, that they are actually arising in response to certain environmental input. And if environment is at the foundation of this whole developmental model, and environment is the thing we can intervene on, then we have a lot of power here. There's a lot more to be done here than we might be thinking about. And there's a lot of interesting literature that kind of hints at that we actually, if we take early developmental influences and environmental influences into account, that the heritability estimates of IQ drop by 20%. We have a lot more power in development than we think we do. And that work that Michael Meany did, I'm sure is familiar to everyone, but just sort of started on this idea that something environmental like the mother's behavior with the rat when it was in that first week of life was having an impact on, I couldn't find, apparently like real rat faces when they're scared are kind of weird looking, so have an impact on this behavioral phenotype of fearfulness and high sensitivity to threat and high stress reactivity. And what was fascinating about his work is that he really laid out the exact mechanistic biological processes by which this environmental experience was cascading out into this behavioral phenotype. And he was not by any means the first person to be demonstrating programming effects, and this was something that's been well understood in biology for a long time, but I think it was profound as psychologists because this is one of the first very clear examples of a programming effect to a psychological trait or to a behavior, that we were talking about things now that were not just physical adaptations to the environment, but behavioral ones. And remember that when we're talking about fearfulness, sensitivity to threat, for those of us in the room, right, like when we study that, we're calling that pathology. We're calling that a problem, a maladaptive trait that we want to intervene on. But why would you want to ever have that rat mom then? What's that? Why is this a phenomenon that is happening? And it may be that, one person's maladaptation is another person's survival, right? This is exactly what Martha was talking about earlier, that there may be a good reason for a mom to be able to communicate to her offspring, if the world is a dangerous place, then I'm going to calibrate that kind of threat detection to preference sensitivity over specificity. It's okay if you false alarm too often. So the idea that these very important behavioral systems in our minds are being calibrated at this early developmental level based on the environmental experiences that we need to know about early that are specific to our environment. Genes are never going to get us there. That evolution doesn't plan that far ahead. There's too much variation from one generation to the next or one environment to the next that we have this mechanism in our developmental biology that is designed to take in information from the environment and develop in accordance with what's going to be the most adaptive response. And like Martha was talking about, I put in my title, I call these adaptations of compromise, because it may be that it comes at a cost. Right? There are, in the school system, in a social setting, having that kind of threat reactivity is going to be consequential. But developmentally, it was a compromise worth making if it's going to have a survival advantage. So it's not to say that it's not something we don't want to intervene on, but it is to say that there is a good, logical, and predictable biological process at play here, and that gives us a little bit of power in generating hypotheses and testing processes and not responding to these things like true pathology that are aberrations or that have gone wrong. So this idea of programming offspring, I think, is an incredibly important process in understanding development, and it is an important phenomenon to recognize that it is not a nature or nurture, or not even a nature and nurture, but that these are actually the same language and the same process. But what's interesting to me is if you think about that rat model, that first week of the rat's life is the sensitive period for this type of input. That's the only time that the brain is open to the maternal behavior. Before or after that isn't going to change anything. Well, if we try to say, okay, well, when is that in a child's life? It turns out that it's in the second and third trimester of pregnancy. And so we have begun to learn a lot more about what's happening during pregnancy and that mechanism of communication, because it's not going to be physical care, obviously, right? But it is going to be many other, did something just bark? No, it's just me. But there are other types of processes that are happening. Maternal, I can't read my own slide here, nutritional insufficiency, maternal health, and environmental toxins, and all of these things that are going to have direct effects on the developing nervous system, but are also going to be funneled through this HPA system, which is a very intricate dance between the mother and the offspring in terms of cortisol reactivity in both the mother and the fetus and the placenta, and all of this communication that seems to be a substrate by which mom can send signals to say, there is threat, there is challenge in this environment, and you need to be developing in accordance to that. So what I want to talk about, I guess I'll mention that there are a lot of different things that we know are going to be affected by cues of stress during gestation, and there's some evidence around behavioral things like sensitivity to threat detection, that amplification of fear conditioning, conditioning to fear in fewer trials or more sensitively to different types of things in the environment. But there's also some physiological mechanistic programming effects that are happening prenatally. And I want to talk about this for the rest of my time just in terms of a point in development that we can get perhaps a lot of bang for our buck in terms of intervention, and a point in development where the structural inequalities in society are pretty rampant and substantial because it has so many broad implications for health. You want to talk about the obesity epidemic? This is another factor that's going to play into whose metabolic system is set to not work well in an obesogenic environment, or vice versa. I have underlined here this dopaminergic decision-making profiles only because that's what I do. That's all I'm going to talk about in this context. But I am interested in these early developmental influences in that process, and the idea that that as a phenotype, as a behavioral trait, is being programmed at an early stage as an adaptation that might come with risk factors associated with it. So what are the implications then for policy? And again, this is where I veer off a little bit into not exactly what I do, but I think what should be mattering to us most at the moment. And one of them is environmental protection. This is not the time to be scaling back at the EPA. We are really in a bad place with a tremendous gap between what we know scientifically and what we are doing about it. There's sort of changes that can be implemented at this point that don't need any more science. We have to get lead out of the environment. We cannot have it in the drinking water. It's not okay. We know that. But we have to understand the magnitude of what these factors mean for development, because there's a lot of mechanisms in which there may be intergenerational transfer that we are not accounting for. So we have a situation that always kind of irritates me, where we have polluted our oceans to the point where our solution is to tell pregnant women that they shouldn't eat certain types of fish. That seems offensive to me. Clean up your ocean, don't pollute. Like, if we can't eat certain types of fish, something's wrong. But the reason why it's certain types of fish and not other fish is important too. So it's a function of bioaccumulation, right? If being in the ocean means every fish in the ocean gets 1 unit of mercury, the big fish that eat all the little fish have their unit, and they get the unit of every other fish they eat, because it is bioaccumulating, because it is not excreted from their bodies. It's stored in their bodies in a way that then just builds up. Guess who else is a big fish in this eating environment? Humans are, right? So any type of bioaccumulating toxin develops, it kind of stays with us over time in a way that could have important implications for when it gets passed on to other environments. And so there was one study that I thought was particularly disturbing, that some biologists had done a study of umbilical transfer of PCBs that were in a contaminated lake. That a particular community relied on this lake a lot for fishing for not only their industry, but ate a tremendous amount of fish from this particular lake. And what they were able to estimate is that the volume of this contaminant that would get into the human body and stay in the human body, assuming that that woman who had that contaminant level became pregnant and breastfed that baby, and that baby were a girl and carried on that pregnancy and breastfed, that there would be contaminant measurable 5 generations out, even if we didn't introduce any further contaminant in the environment. If they stopped eating fish today, we can still see evidence of that 5 generations back. So we take that example for lead. We had a lovely talk today about bone health. Lead is another problem for bones, right? So if you're exposed to lead while you are developing all of that bone tissue, Lead takes the place of calcium in that bone structure and will get stored into that infrastructure in your body. It's going to be stored in your teeth and bones, and we know it doesn't go away. It's still there over 30 years later. It's released in response to stress, but also if you've got a pregnant mom who isn't getting enough nutrition to be able to support her own health and the growing fetus, guess what? The body will go into her bones and teeth and try to pull that calcium out to give to the baby. And if it goes in and it finds lead, it's pulling that back out, right? So if we don't just have a group of kids in Michigan right now who grew up drinking leaded water, we have a generation of kids who will not be born for 30 years who are already at risk because of this contamination. So for all of you graduate students in the room, you've got, the clock's ticking. You've got to solve a lot of problems before those kids are born, because there may be some risk mechanisms at place that even if we act now to clean up that exposure, we still have to recognize the consequences are coming down the pike. Smoking cessation is another one of personal interest to me, and that we're looking at. Nicotine is a behavioral teratogen, and because it mimics acetylcholine in the brain, it is exquisitely sensitive to brain development and is going to affect brain development at doses well below what it would take to affect physical growth. So if you see on those packs, the only thing they're warning pregnant women about is that it could affect low birth weight. If it's gone that far, it has affected brain development, and it's going to affect brain development at much less. Nicotine, another thing that we're starting to learn about, we sort of realized that secondhand smoke was a bigger problem than we thought about. So-called third-hand smoke, the residue, the physical touching stuff that smoke sort of settles on, is still a significant problem for exposure to kids. So it doesn't even mean that the mom has to be the one smoking. The environment can be dangerous for them, and in low SES environments, that's even worse. Crowded public housing where the ventilation, it can be a neighbor who's smoking. Waitresses who are working in kind of lower SES jobs where smoking's allowed in restaurants or those places, they're exposed at a higher rate. And if you think about those epigenetic risks, so not just the direct programming risks of the nicotine, but the effects of nicotine exposure on epigenetic methylation, we again are looking at the risks for intergenerational transfer that are going to cascade so far beyond that when we think about making a policy change, we can't expect to see our investment returned immediately. We have to know that we're in the long game, and we have to be playing that long-term investment to be looking at whether or not there's efficacy and kind of having faith that there will be. I am going to just probably scroll through just tiny very quickly because we are out of time. I want to keep us on time, but I do want to acknowledge that I've really kind of focused on the prenatal period because I think it's an area or a developmental period that is really interesting to me and really important and really, I think, under-identified. But of course, many of the other people who have talked today, I've highlighted the other very important periods of brain development and sensitivity. So it's certainly not the case that we lose our chance if the baby is born and that we don't have other opportunities or important stages to intervene at. And certainly puberty is another one that Laura talked about. Parenthood is one that I think is interesting. So again, huge hormonal shifts that are going to reorganize brain processes and priorities, that it may be a sensitive time to intervene to help parents during that transition to kind of optimize their own parental behavior. And just sort of generic summaries about the idea that these programming effects present unique challenges in terms of trying to identify that causal link. So what Laura talked about earlier, that researchers would say, oh, osteoporosis, why would you study adolescents? We sometimes have a hard time selling our science when we're saying there's a 40-year gap between the risk and the outcome. But we know as preventionists, right, that earlier the better, and that can be hard to sell. And how do you design a study that says, I want to show you that I did what I told you I was going to do, when what I'm trying to do is reduce your chance of dying at 50 from a heart attack by changing the nutritional quality that your mother has access to when she's pregnant with you. these types of health connections are real, and we know that scientifically, but it's a hard sell that we have to figure out how to make that translation over decades of time and generations of individuals, and that is all I'm going to say.
Thank you. And so moving us right along, we have one more speaker as part of this session. That will be Dr. Idam Shalem, who joined the Department of Biobehavioral Health here in 2014. And he will be speaking about the biological embedding of the diversity.
So thank you, Hannah, for the introduction. Thank you, Danny and Emma, for the invitation. First of all, heads up, I'm recovering from a cold, so if I'm starting to cough in the middle, I apologize in advance. As Hannah mentioned, I'm going to talk about the biological embedding of real-life diversity. As I wrote in the abstract for this meeting, the goal of this talk is to discuss the current state of the science of the biological embedding, of how early adversity gets under the skin to influence adult health and disease. So if you want to evoke change in the health trajectories for survivors, we need to fill gaps in understanding of this biological embedding and to further explore also mechanisms for resilience. So what I'm going to discuss today, I'm going to present two research examples, first using telomere biology and also using dynamic gene expression changes as potential platforms for both discovery and intervention studies. So first I want to start with three important frameworks in relation to biological embedding. So the first one, where is Lisa? It's familiar, right? Yeah. So the first one is the developmental origin of health and disease that suggests that the biological embedding can begin already in utero. So the Dohan model, also known as the Barker hypothesis, suggests that adverse conditions during pregnancy, which can obviously influence the environment of the fetus, will have long-lasting effect on adult health, including increased risk for cardiovascular disease and metabolic disorders, as well as early mortality. So in other words, the Dohan model suggests the hypothesis that impaired growth in utero, whether it's caused by maternal psychological stress, depression, poverty, malnutrition, etc., can again increase the risk for later life diseases, probably mediated through stable epigenetic modifications. A second model that is important to consider is this CDC experiment from the CDC Adverse Childhood Experiences study. And this model proposes a whole life developmental cascade leading from adverse childhood experiences to social, emotional, and cognitive impairment, adoptions of health risk behaviors up to morbidity and early mortality. Even though these studies consider to be a landmark in epidemiological research, it also highlights some scientific gaps. And in particular, they're missing the biological embedding mechanisms that can explain some of the links that you see here in this pyramid. And lastly, the third model that is important to consider is Bruce McEwen's allostatic load. Some speakers already mentioned this model before. So in this model, the perception of stress is influenced by one's genes, experience, and behavior. In particular, also highlights some of the environmental exposures, trauma and abuse, as you can see at the top. Overall, this leads to allostasis and adaptation, but over time, the overexposure to stress mediators will cause this allostatic load, which will cause wear and tear of physiological systems, and again, can increase the risk to develop diseases later in life. This is also an important model because it also highlights some of the biological mechanisms. In particular, McEwen is talking about the neuroendocrine and immune systems as part of the processes that can mediate these effects. So another factor, a more recent and novel factor that can also provide some of the explanation for these biological mechanisms linking stress to disease is telomere length. So I assume many of you heard about telomeres, but if not, so telomeres are this repetitive DNA sequences and proteins that are found at the ends of all chromosomes in all of our cells, other than red blood cells. And they form this cap end structure at the end of chromosomes, protecting the chromosomes from DNA damage response. And the best way to think about it is this analogy with the plastic tips at the end of the shoelace that protect the shoelaces from fraying. So this is very similar function that telomere play on the end of the chromosomes. Now the problem is that every time the cell is dividing, we are losing small segments of telomeres until they become critically too short, and that signals the cell to stop dividing. This initiates DNA damage response, and eventually the cell enters a state of senescence and replicator arrest, which is one of the hallmarks of aging. So this is how we age basically at the cellular level. By far the biggest predictor of the length of telomeres is chronological age. The older we get, the shorter the telomeres are because our cells are dividing many more times, especially our stem cells. But research from the past 10, 15 years also provide evidence that environmental exposure, especially toxic stress in your life, can accelerate the shortening of telomeres or accelerate the biological aging of young individuals. So this figure illustrates a conceptual model of the influence of lifestyle influences on telomere length. So we begin with the prenatal and childhood adversity that can influence the rate of telomere shortening. We believe this goes in one way, hence you see these unidirectional arrowheads. And later in life, there is evidence of associations between mental health disorders, specifically depression, as well as physical conditions with shorter telomeres. Cancer is also linked to short telomeres. And there's also studies indicating that short telomeres can predict early mortality. And as you can see at the top, there's also evidence that healthy lifestyle factors, specifically exercise, mindfulness, meditation, healthy diet, social support, all of these factors can mitigate the effect of stress on the rate of telomere shortening. Here specifically, I'm going to focus on childhood adversity. So what is the evidence that childhood stress or maltreatment is related to short telomeres? So this is the first study that provided this evidence, published in 2010, just to show you how new this field is, by Audrey Tirka and colleagues, providing preliminary evidence that adult participants who retrospectively recalled incidents of physical and emotional neglect had shorter telomeres on average compared to the control group, independent of age, sex, BMI, and smoking. Many more studies followed, providing more and more evidence that early trauma is related to short telomeres. This last study that you see here at the bottom by Stacy Drewer and colleagues was the first study that was published in relation to children, showing that greater exposure to poor institutional care was also related to shorter telomeres. But even though all these studies provided some evidence, it still possessed several limitations. Mainly that almost all the studies have looked at adult populations who retrospectively recalled incidents of abuse. They relied on self-reports, and they all used cross-sectional designs. They measured telomeres at one time point. And with telomeres, you really want to look at change over time, so you want to have this prospective design when you collect multiple samples over time. So we tried to overcome some of these limitations in a study that we published several years ago. The participants in our study came from the Environmental Risk or E-Risk study, which is a representative birth cohort in the UK, more than 1,000 twin pairs. You see at the bottom the study design, the collection phases are circled in blue. So we had the first DNA that was collected at the age of 5, and then the second DNA that was collected at the age of 10. The study is still ongoing. We finished the data collection at age 18. We're currently analyzing the telomeres also at age 18. But what I'm going to show you is the telomere change from 5 to 10 in response to violence exposure. So we also prospectively assessed violence in children, specifically domestic violence between the mother and the partner, frequent bullying victimization, and actual physical maltreatment by an adult. So these were the main findings. We found a main effect of the cumulative exposure to stress. And this is also based on evidence that the increased exposure to stress, this chronic stress, also based on the CDC ACE study, that multiple exposure to multiple incidents is confer greater risk. This is exactly what you see here. Only the children who were exposed to 2 or more types of violence had the fastest telomere shortening from 5 to 10 years, compared to children who were exposed to one type of violence. This is the broken line, and at the top, kids were not exposed to stress at all. So this study was important because this is the first evidence that violence exposure can be related to telomere shortening already in childhood, before the onset of age-related diseases. So provide potential mechanisms how telomeres can explain this biological embedding of early life stress. So speaking about the mechanism, this is where it's becoming very complicated. So again, if we take the example of early abuse, we know it's a risk factor for many adult diseases. including cardiovascular disease, metabolic syndrome, cancer, early mortality, mental health conditions. So it may be that telomeres can act as a master integrator, linking this exposure, toxic exposure in early life to later life diseases, but we still don't really understand the mechanisms, how stress is causing the shortening of telomeres, or how short the telomeres may cause the onset of these diseases. There's still a lot of unknowns. We think it's mediated through biochemical stressors in the cell and also through epigenetic modifications, but obviously it's much more complicated than that. We know it's also mediated through the stress response system, specifically through the HPA axis. Telomerase enzyme, this is the enzyme that can protect and lengthen the telomeres. It is also implicated in these mechanisms. Inflammation and oxidative stress are very important factors, mechanisms that can also regulate telomeres, and also mitochondrial regulation. So all these factors we're currently investigating in the lab. We're trying to understand the factors that can regulate telomeres and as a potential target again for intervention. So another general regulator that can actually link all of these factors that you see here together is by investigating gene expression. And the reason why gene expression is also very appealing in relation to translational research is these studies that show that specific gene expression signatures can predict clinical status and specific outcomes in breast cancer. And you can say that these studies are fairly old, even though they were published in PNAS, Nature, and New England Journal of Medicine. But here's a more recent evidence published last year, again showing how specific gene expression signatures can also help clinicians determine risk in leukemia. So at least in cancer research, there's evidence for the utility of using gene expression changes. Now, the problem in doing this kind of research is that gene expression is very noisy. It's also influenced by many factors. So if you take two groups of early adversity and control groups and you draw blood and you look at gene expression, this will not be an ideal design to look at differential response. So you want to use a specific design to test that. So this is what we did last year. We ran a small pilot study. And this is the conceptual model that we developed. So basically, we are using acute stress response in the lab to measure changes in gene expression. And what I'm showing you here is that acute psychological stress will induce system-specific gene expression changes, which in turn will cause metabolic, immune system, and DNA damage responses. Again, that can influence the gene expression responses. This by itself can cause telomere shortening, senescence, and apoptosis, which can increase the risk to develop specific diseases if the stress is repeated. And specifically in the study that we did, we wanted to test this box here, and we hypothesized that this response will be dysregulated or programmed in individuals who were exposed to early adversity. So what we did is we exposed individuals who were exposed to early adversity to acute stress, and we wanted to look at their gene expression compared to people who were not exposed to stress. So let me show you the methods for this pilot study that we did. We recruited 12 male participants between the ages of 18 to 25. We focused on men in this pilot study because of the unknown sex differences in response to stress and the small sample size for stratified analysis, but we are obviously interested to increase the sample size and include women participants as well. In order to obtain the risk group who were exposed to adversity, we used this stressful life event screening questionnaire that is a 13-item self-report that assesses exposure to trauma in their life up to age 18. And here are some of the examples for adverse exposure in a risk group. And these are pretty significant adversity that our participants experience. And we required that in order for them to be included in the risk group, they had to indicate at least 3 of these exposures. So we had a pretty extreme risk group in our study. And we also used the screening questionnaire to screen participants who were not exposed to traumatic events in early life. So altogether, we had 6 participants who were included in the risk group and 6 participants who were included in the control group. And we used this within subject randomized design, and we included 1 week the acute stress. We used the Trier social stress test, or the TSST, to induce stress. And 1 week there was no stress resting condition. They basically sat in a room and they didn't do anything. And we obviously gave them a lot of questionnaires, and we collected saliva samples and repeated blood samples to look at gene expression changes over time. So let me jump directly to the results, and what I'm going to show you, this is the results of the salivary cortisol, just to show the effect of the stress compared to the resting condition. So this is for the entire sample within subject analysis. And what you see here, the red, this is the acute stress condition, the TSST, versus the no stress condition. So what this figure is showing is that the acute stress that we did was stressful enough, and we detected significant cortisol changes when compared to the resting condition when people are resting and are not stressed. Then when we stratified by risk group, we saw that this response to the TSST, to the acute stress, was specifically in the risk group. So the risk group exhibit these increased physiological reactivity through a higher salivary cortisol level compared to the control group. This is what you see here on the left. Oh, you can see this. Sorry, guys. So what you see here on the left, this is the risk group, and this is the control group in the TSST, and this is the resting control condition. So in the resting condition, there's no differences. So this is our another control that we included. And overall, what we are showing here is we're confirming previous studies showing that early trauma through this programming of biological systems can dysregulate the HPA axis to show higher levels of salivary cortisol. So what about the gene expression results? So what I'm going to show you is a preliminary prototype looking, first of all, at one candidate gene, specifically the glucocorticoid receptor gene. And again, I'm going to show you the results for the entire group, for the 12 individuals. And again, looking at the TSST in the blue line versus the no stress condition. So again, like with the salivary cortisol, what you see here is when you induce stress in the lab, you see an activation of the gene expression for the glucocorticoid receptor, which is an adaptive response. You want to increase the level of this gene in order to shut down the HPA axis, so this is considered to be an adaptive response. And this design also showing that we can also look at recovery processes. So I didn't mention that before, the salivary cortisol we looked at 90 minutes post-stress. But here we're looking at gene expression. The first one is 30 minutes before the onset of the stress. And the last one is 4 hours after stress. So we have 4 repeated measures up to 4 hours after the stress, and you can see this increase and decline in the levels of the gene expression for the GR. And then what happened when we stratified it by risk group, we see that this response to the acute stress was specifically in the control group. Again, I mentioned that this is an adaptive response, and you see that indeed in the control group. In the risk group, in this broken line, you see a blunted response. They were unable to increase the level of the GR in response to acute stress. And notably, there were no differences, no significant differences in the resting control condition. Again, we wanted to include this control condition because gene expression is noisy. And here we're showing that there is some noise over time, over 5 hours, but there is no significant differences. So these findings provide a proof of concept for this design and as a platform for discovery of other potential genes. But it can also help to explain some of the mechanisms that actually Lisa mentioned before. So we know that from previous studies, Michael studies, that stress in early life can cause hypermethylation of the GR. This will in turn cause lower expression level of the GR, specifically in the hippocampus, which will reduce the ability to shut down the HPA axis, which will cause higher cortisol response that we see actually in our study. So this can also provide some very nice mechanistic story. So in order to understand the full story in our study, we will also going to look at methylation of the GR. So to conclude, what this study is showing is that these preliminary findings suggest that early life adversity can program biological systems. Sorry, I'm half blind. As evidenced by this dysregulated cortisol and gene expression responses. And even though, as I mentioned before, these response patterns can be adaptive over the short time, if they are repeated over time, they can provide this evidence, this mechanism for increased susceptibility of diseases later in life. So what are the next steps? What I showed you was just one gene, candidate genes, but we recently finished to do the sequencing for the entire genome. So we're going to look at the RNA sequencing. We didn't really finish it. Our genetic core facility did the work. But we currently received the data and we are analyzing the RNA-seq. So we are hoping to discover more specific networks in relation to this early life adversity. So this is this RNA sequencing. In this study, we also isolated specific cells, and we are very interested to look at specific types of cells. The results that I showed you was from PBMC, these common mononuclear cells, but we also isolated T cells and hematopoietic stem cells, which is very interesting also in relation to telomere biology. We're also going to measure in this study. And as I mentioned before, we're also going to look at epigenetic modifications down the road, again, to get the complete mechanistic story. And lastly, just going to throw it out there, I probably don't have enough time to talk about it, but this is also something that we are considering to do next year, do this paradigm of parabiosis. So specifically, we're going to use this human-animal interface. This is sort of like reverse translation. What we are proposing to do is to investigate plasma from the risk and control group and to inject that plasma into mice and to see how that influences aging and health processes in the mice. I know it sounds a bit creepy, but people actually doing this work. But this is another types of kind of a novel method to investigate biological embedding and to increase our understanding on specific mechanisms. And I just want to finish, acknowledge all the people who were involved in the studies that I've showed you. This is obviously team science, and to thank you for your attention.
So at this point, I would like to invite all three speakers back up. We'll have a short 8 to 10 minutes of questions.
I have a question for John. Is this on? Yeah. Okay. I'm confused about your TSST findings because
which one, the cortisol?
Yeah.
Okay.
Yeah, the cortisol, and because you should have a response. You should have a cortisol response during the TSST. So I'm curious why you're linking that to the stress, to the life stress.
We definitely showed there was a significant response to the TSST, but when we divided it to the control and the risk group had much higher, significantly higher, it was trending to significance, higher cortisol than the control group. So both groups experienced the stress, but was just higher in the control in the risk group.
Right. Okay. So you would say they were hyper reactive, hyper responsive. Okay,
Yeah.
that helps. Okay. I warmed you up.
I have an observation, I guess, that Lisa and Martha both talked about the adaptiveness of these responses in harsh circumstances where it's necessary to take on some of these behaviors that might be maladaptive in another setting, but are very, very important. You know, it's very much in line with, now I'm forgetting the name of the person, is it Bruce
Ellis?
Ellis.
Yeah.
Del Judas.
It's very much in line with his modeling, and so I guess, you know, what's concerning is people who hear that, who are not us, might think, well, okay, then it's adaptive, and so we don't need to address it. They're adapting to their setting, but I think in prevention science, what we tend to focus on is fixing the damage and reducing the adverse consequences of really negative exposures. But I think what your research, all of your research, points out is how important it is to reduce the exposure in the first place, not just to try to fix things after the fact. So even though we're working hard to try to help children be more resilient in these horrible circumstances, we need to reduce child maltreatment. We need to reduce poverty, and that's where the policy comes in. And why it's so important that we address the roots. And I love your social justice model because you're really looking at the roots and then trying to find acceptable, legitimate channels for that sort of behavior that can actually, it doesn't suppress the survival skills that they've learned, but we find alternative activities for it so that it can be very, very adaptive and, uplifting to the children. I just think that's great. I'm dying to hear how these models affect physiology and so forth. I just think it's compelling from a policy perspective, and I always say this, that if we can show that it's changing neurophysiology that policymakers might pay attention. Because you can see you're changing behavior, you're changing brains, they're just not impressed. But if you tell them, look at these pretty, pictures with the blobs on the brain, and you've got fewer blobs over here, they're different colors, and so that means that you're actually changing human development. Then they do listen. So if for nothing else, that research is so important.
Just one brief, quick comment about this adaptiveness. So Bruce Ellis, for example, I think had a paper published very recently. I think it was Current Direction in Psych Science. Again, talking about how early adversity child maltreatment can produce all of these adaptive responses in the short term, increases cognitive abilities even. But the question is, what is the cost? So there's a trade-off. So they're all using this evolutionary perspective life history model, and they're talking about, yes, adaptive in the short term, what happens in the long term? So that's also a question why it can be beneficial, but then down the road.
Well, and the way we think about it is that, again, like those functional adaptations may still be very useful in certain settings. And so we try, our motto is to meet kids where they are and then help them grow new capacities so that they can adapt to a variety of contexts. Because in essence, they need to be sort of super functional, hyper functional. They need to be able to function in crappy environments and good ones. Yeah, but so life's harder. We already knew that anyway, right? So.
But it's harder for those that are less advantaged.
Yeah.
Obviously.
Yeah. Jose. Oh, sorry, Hannah, you're supposed to be. I just take over everything.
So this, I think, to the whole panel, but I think one of the things that your talks sort of point out is that role of systemic problems, like to the point of, I feel like, it's like in your talk, I feel like it reminds me of when I talk about health disparities sometimes, that at the end of it, I'm just like depressed. And it's very, very disheartening, right? And I think one of the things that makes it challenging is the systemic change piece, right? And, so you've talked about some of the ways in which you can have, try to help the individual maybe influence the system a little bit, like in your intervention, but any thoughts on delivering a system intervention? Like, is there, I don't know if you've thought about that, or, obviously that's sort of like the big, especially if you think about social justice, like, how can you deliver a system level intervention? And just wondering about whether that's something that you thought about, and any thoughts about that? Sorry, it's a big one.
Yeah. By system level, do you mean
Like not
policy
necessarily
kind
through
of?
the individual, I think.
Yeah.
Yeah.
Well, I think that's something we need to think about in terms of what is the scale of the intervention. And I think there's policy opportunities at infrastructure levels, but I also think then we need to harness where are the time points in life that we have access to people, that we can improve broad functional skills that we're not really delivering. So I think PATHS is a good example of that, where we've got schools as this amazing opportunity that kids are required by law to be at. So we maybe see them when they're born in the hospital, and society doesn't have a handle on them again until they're 5. But at that point, or if we chose to go earlier, we could be doing more with that time than just ensuring that math and English and writing get done, right? So if we're actually take it upon ourselves to say self-coping and emotional regulation are developmental milestones that you have to achieve as a human just as much as math and writing, then we could be thinking about more established infrastructure of that being a curriculum goal, that is something that we actually do. I have colleagues who've talked about, we could be doing more with teenagers to actually talk about healthy relationships and training that than just saying don't have sex or have safe sex, or that there's what are the things we need to be functional at in life, and where are the places we could actually make it society's job instead of the parents' job in all cases.
Well, and I know we need to shut down, but just I think, again, getting back to George Albee, that one of the things that we can do is embed better education around things like racism and
Yeah, that's another
you know,
one.
acceptance of inequality as being okay.
Yeah.
And we, you know, and because it will take probably, you know, societal change for the highest levels to occur. And I don't know that we as psychologists are equipped to develop an intervention for that. But what we can do is educate kids better and create a better, more human populace.
And this gets back at that, the long game problem, is
Yep.
that major investment is so that it'll pay off in the next generation, and that's hard to sell.
Yep.
So just to, sorry, I'm But just these are questions that the center and the PRC is addressing in terms of systems change, and has developed, you know, infrastructure of system change across the state of Pennsylvania. And in the coalition, we're working on system change where, and not that it's happened, but for, you know, in a lot of the places that we're looking at, like in Baltimore City, to get all the systems involved to actually, you know, have a continuum of care that attends to all the various needs of whatever developmental group you're looking at, or all of them really. Mental health, you know, getting mental health systems involved with juvenile justice and with the school systems, and you could go on and on with all the different institutions that need to all be concerned in a coordinated way with children's healthy development. And so there are some things going on in the background, and looking for foundation funding from like Arnold that wants to institutionalize these kinds of systems change. It's a very big foundation, and it's a huge monumental job, but there are these things bubbling up to do just that.
Thank you very much, Anne.